So this is a continuation of our webinar series from last year on eDNA. Organizing this with us is Susie Theroux from Southern California Coastal Water Research Project. So I'm going to let Susie take over and introduce our outstanding speaker for today. This meeting is being recorded. We will have it posted on our YouTube channel, hopefully by week's end. We've had a bit of trouble with our editing software. It's in the cloud and connections haven't been that great, but we hope to get it up very, very quickly. If you have questions, please hold them for our presenter until after the presentation has been given. You can also send them via the chat feature. Thank you again for joining us.
Webinars is available on the Monitoring Council's Collaboration Network website, so you can go and rewatch any of the previous webinars that we've had. We'll have 2 more on deck coming this year to wrap up the seminar series, one on eDNA for human health and then one on data visualization and analytical techniques. So be on the lookout for the announcements for those ones. Today, my goal is really just to provide a brief introduction, kind of bring us all back up to speed. And if you've missed any of the previous eDNA 101 webinars, it just gives you a little bit of a foundation for what we're going to be talking about today with regards to eDNA for species surveillance. And then I'm going to hand it off to Dr. Sarah Brown from the California Department of Water Resources, and she's going to be telling us about the great research that they've been doing there to develop novel analytical tools for detecting targeted species of interest out in the wild. So with that, let's just make sure that we're all talking about the same thing when we talk about eDNA or environmental DNA and what exactly it is. So I like to use the most broad definition of eDNA, which is really just the DNA that we found out in the environment. And it's often entering the environment by the shedding of skin cells, or maybe it's mucus or saliva, and it's coming from organisms that are transiting through the environment, everything from microbes to turtles to whales. Oftentimes when we're going out into the field and we're collecting these environmental DNA samples, we're maybe taking a liter of water or we're taking a soil sample, we're bringing it back to the lab. We're going to extract the genetic material away from all the other cellular debris and environmental constituents, and we are left with a pure DNA sample that then we're going to subject to some analytical techniques like DNA sequencing or quantitative PCR. For a little bit of background on what some of these different analytical techniques are, I like to talk about them kind of in 3 main buckets. The first one in the eDNA toolbox is a quantitative approach, which might be quantitative PCR or digital PCR. And you might be familiar with quantitative PCR, of course, from all of our COVID tests that we've had. And this is a really great approach if you're looking to quantify a number of target gene copies that are present in your sample. So maybe that's because you're looking for harmful pathogens in a beach water quality monitoring sample, or maybe you're looking for a number of microcystis cells that are present in a lake sample. If you're instead interested in identifying who an organism is that you have in your sample, maybe you caught a fish, you're not exactly sure what species it is, you might use a DNA barcode sequencing approach where you might take a swab or you take a fin clip, and then you're going to send that off to a sequencing facility so that you can generate a reference sequence and get a better sense of who taxonomically that organism is. If instead you want to look more broadly at all the biodiversity present in your sample, maybe you have a lake or a river sample and you want to see a snapshot of all the different fish species that are present, you would use what we call DNA metabarcoding approach or DNA fingerprinting, where we're using short snippets of DNA to identify the different species that are present in our sample. What we'll also be hearing about today, though, is some of the more novel techniques that allow for rapid field-based identifications that are even building out this eDNA toolbox and giving us even more opportunities for rapidly and sensitively detect species in the environment. I wanted to highlight a couple of well-known examples of where eDNA-based approaches are used for invasive or endangered species monitoring. One of the earliest examples is looking for the invasive Asian carp in a series of waterways in the Midwest in the United States. This was one of the first applications of quantitative PCR type of approach for detecting rare aquatic species and then implementing management decision-making based on where those species were observed to be. Another good example is looking for the invasive green crab up in the Puget Sound, where they've been coupling some traditional visual-based or trapping-based approaches with quantitative PCR-based approaches using a species-specific eDNA we can
Susie, yeah, your voice is breaking up. We may have lost you.
I will maybe turn off my camera, see if that improves our connection. Can you hear me now?
Let's hope so.
Okay, I'm almost done, and then I'll hand it over to Sarah.
Thank
and
you.
the last example, yeah, I was going to give was in great crested newt monitoring in the United Kingdom, where there's been a species-specific quantitative PCR probe developed for monitoring the great crested newt, and it actually has been implemented in a broad-scale biomonitoring program where there are now lab accreditation programs and proficiency testing schema that have been developed around this great crested newt monitoring. And now the observations of great crested newt based on eDNA detections can be used for environmental management decision-making. With that, I'm going to skip ahead so that I can hand things over to Sarah today, who's going to be telling us even more about invasive and endangered species monitoring work that they've been doing in the California Department of Water Resources. And I'm really excited to hear about all of the novel techniques that they've been developing to help expand our eDNA toolkit.
Okay, thank you so much, Susie, for that introduction, and for Eric for inviting us and providing this forum. I'm so excited to talk to you guys today about the eDNA work that we're doing in collaboration with UC Davis and with our brand new genetics lab at the Department of Water Resources. Got some exciting stuff to share with you. And so just real briefly, just the outline here, I'll be talking about GWR operations. So our endangered species protections and monitoring programs, the use of eDNA for surveillance, for species surveillance, and the use and management. And then we'll go into some case studies for using eDNA to monitor invasive species and endangered species. So as many of you know, the Department of Water Resource delivers water to Californians. We're responsible for managing the state's resources, operating and maintaining the state water project, protecting and restoring the Delta, and delivering water to 27 million Californians and thousands of acres of farmland. This water travels from streams and rivers to reservoirs and canals and ultimately to your faucet. So not only is DWR tasked with operating the state water project and getting water out to Californians, we are also tasked with operating all of our systems in a manner that protects species. And so you can see from the previous map, or the map in the previous slide, that the state water project pulls water in from diverse habitats all throughout the state that contains many species at risks. And so the state water project is operated in a manner that protects species and that critical habitat. And so we operate under the State and Federal Endangered Species Act and in compliance with incidental take permits and water rights decisions. And so we ensure that water flows to aid fish survival and to minimize, avoid, and fully mitigate the impacts of those operations on fish at risk of extinction. So we do a lot of monitoring at DWR, and we have a lot of traditional monitoring of species. And so this work involves going out and capturing individuals that we count. And there's many ways that we go about this, and that's through various nets, rotary screw traps, pike traps, and trawls. And so, like I said, this requires the actual physical capture of individuals. And for rare species, that might not be the best way to try to monitor them and to determine their presence in the system. So we need a tool in the toolbox that has increased sensitivity to detect these rare species. in comes eDNA, as Susie so greatly introduced. The eDNA is a tool that essentially contains a snapshot of the community of organisms that are in the water sample. And so these applications of eDNA are wide-ranging, including species detection, biodiversity surveys, and assessment indices. It's an incredibly sensitive approach. We don't need to capture the individual species, so there's no potential harm to them. There's a potential reduced cost as far as field work and stuff goes. And in some cases, we can have an increased number of species detected, like with the meta-barcoding approach that Susie mentioned. And eDNA, and I'll be speaking about eRNA, that degrades pretty quickly, so it can provide a current snapshot of the community of interest. So just some of the applications of eDNA are monitoring for rare and invasive and nuisance species, looking at biodiversity and climate change, and then pathogen monitoring as well. And I will be focusing on invasive species first, and then rare species monitoring. So invasive species are costly in terms of the environmental damage to state and federal budgets. And they're also very expensive to try to eradicate. So the earlier an invasion can be detected, the better it is for preventing damage to the environment and for the pocketbooks of the state and federal governments. And so eDNA is a great tool to be able to monitor and detect invasions and to assess the eradication efforts that agencies have been conducting. So one of the projects that we're working on in collaboration with UC Davis is using eDNA and eRNA to assess eradication efforts of nutria in California. So nutria are relatively new invasive to California. They're originally native to South America. They burrow into levees, causing them to breach and weakening structural foundations and eroding banks. Additionally, they remove stabilizing vegetation and are carriers of diseases. There was a reproducing population in the San Joaquin Valley as of 2017. About 3 million is spent annually on trying to eradicate and manage for nutria. To date, I think there's about 3,700 that have been removed. I'm sure that number has increased. And so, like I said, California is working on trying to eradicate and contain the populations. So the, like I said, nutria are present in California waterways, but this is still at the early invasive stages. And so there's still work that we can do to help get rid of them. And I just want to highlight these pictures here. So this picture on the left is from a normal marsh in the Chesapeake Bay in 1939, before nutria were introduced in the 1940s. This picture on the right is from 1989, where over 50% of the marshes had been converted to open water due to the destructive feeding habitats. So just an example of how destructive nutria can be to the environment. And so they are concerned to DWR for the potential damage to levees. And so we are using a new technology that Susie mentioned, called Sherlock, that we can use both eDNA and eRNA to detect and locate individuals quickly. One of the benefits of eRNA is that it can detect the most recent activity. And so eRNA degrades more quickly than DNA. It can persist outside of an animal for about 13 to 24 hours. And it's about days or weeks for DNA. And so this is great as far as being able to detect recent activity of nutria when we are trying to eradicate them. We can see how effective our eradication efforts have been. So just a brief introduction to what Sherlock is. So it's based on the CRISPR system, which utilizes enzymes from the bacterial immune system. And so Sherlock has many qualities that lend it to genetic monitoring. It can be performed at a single temperature, unlike traditional PCR that requires a thermal cycler. It requires multiple temperature steps. And so because of the single temperature reaction, it's suitable for field deployment. Additionally, it's fast. It can be has multiple steps that can be completed in just one tube. And it's incredibly sensitive and accurate. So just briefly, a little bit of genetics here. So the DNA, we have DNA input, and let me see. The DNA is amplified and transcribed to RNA by a recombinase polymerase amplification, which again is performed at a single temperature, so 37 degrees Celsius. That amplified RNA then is bound to guide RNA, which is the sequence of interest, so the mitochondrial snippet from the nutria. And then an activated Cas13 cleaves an RNA reporter that has a fluorescence molecule, and that essentially releases light to our fluorescent reader. That the fluorescent reader is able to record that emittance of light. So we have just some preliminary data to share. Again, like I said, we've been looking at studying the DNA and RNA detection with these tests. And the chart up on the top shows essentially the whether or not the off target. So the off target is that maroon line down at the bottom, and the target, which is nutria DNA, is up at the top with kind of the yellow box. So just showing that we are amplifying nutria DNA and no other rodent species or any other species for that matter. And then down on the bottom is another chart showing essentially that we're able to amplify nutria RNA pretty rapidly and very quickly. So this is very exciting that we're able to amplify nutria and not amplifying the off targets. And so we have a lot more work to do on this project to test. So that was all done with just nutria DNA. And so now we're moving on to tests with eDNA. And so we have a lot more work, but we're really excited about moving forward with this. And so, like I said, the goal is to use both eDNA and eRNA to detect not only new invasions, but the efficacy of eradications. And this is just one more tool in the fight against invasive species. And so we have current work, similar work that we are looking to conduct pretty soon that will be on zebra and quagga mussels that will have the same kind of technology of looking at eDNA and eRNA with Sherlock. Okay, and then now we can move over to environmental DNA of endangered and rare species. So delta smelt were previously one of the most abundant species in the San Francisco estuary, and they were listed as endangered in 1993. And some of the threats to them include habitat alteration, altered hydrology, and invasive species. And so when we're monitoring them, some of the needs for detection are kind of twofold, and that we're out in the wild and we need to aid with monitoring efforts. Or additionally, need to monitor near water operations equipment to make sure that smelt don't get entrained in the pumps. So like I mentioned, the delta smelt can become entrained at the GWR pumping facility. And so Clifton Court Forebay, with this big reservoir here, is a reservoir where water enters before the GWR pumping facility. And there are louvers that divert adult and juvenile fish from entering the pumps where they're sampled and then released elsewhere in the delta. And one of the things that's unknown is whether or not delta smelt larvae, like how many of them are getting entrained. And so our goal was to monitor and quantify larval entrainment at Clifton Court Forebay. And so we were going to determine detection probabilities from known biomasses and distances and determine optimal eDNA sampling strategies for field conditions. And I'll just be presenting on some of our lab experiments that we've conducted. The data analysis for our field work is still going on. So our question was, what is the relationship between biomass and eDNA concentration for delta smelt? So we conducted a laboratory experiment where we tried to find the lowest concentration of larval smelt eDNA that could be detected under ideal conditions. So we placed a known biomass, 1 individual, 3 individuals, 10, 30, 60, 100, and 300 of fish into individual tanks of clean water and then extracted the DNA from that water. And so we then used qPCR, like Susie mentioned, a very sensitive way to detect eDNA. And our goal was to try to find the lowest detectable concentration of DNA. So we have concentration of DNA on the y-axis and biomass of larvae on the x-axis. And we detected delta smelt eDNA from all biomasses tested, and the negative controls were negative, so no contamination occurred. And what we find is a beautiful linear relationship that the concentration of eDNA increased with increasing biomass. And we found that variation between replicates was high. And in a laboratory setting, we were able to detect down to 1 larvae, which is pretty incredible, seeing how small these guys are. And like I said, the data is currently being analyzed for a field study where we had a live car out in the West Canal and actually sampled eDNA from the water, trying to detect different biomasses from different distances. And so hopefully those results will be forthcoming soon. So like I mentioned, in addition to getting to concerns about smelt getting entrained in the pumps, scientists with multiple agencies conduct traditional surveys year-round to monitor delta smelt throughout the Delta and the San Francisco Bay Estuary. So we have the U.S. Fish and Wildlife Service Enhanced Delta Smelt Monitoring Team, EDSM. They estimate the total abundance of delta smelt on a weekly basis and help to estimate their spatial distribution. This is a very intensive process. It's year-long, involves multiple gear types, and eDNA could possibly aid in detection. So this project is work that's led by Ravi Nagarajan at UC Davis, and he modified the delta smelt, the delta smelt Sherlock assay so that it would be more sensitive, and tested the sensitivity in the laboratory and found that at low densities, so 124 individuals, and at high densities, almost 900 individuals, that the assay was able to detect them well. And then conducted co-sampling with the EDSM monitoring team with eDNA in addition to their gear-based sampling. and found that that 3 eDNA samples collected in 2021 in the lower Sacramento River. So these blue dots here on this map showed that delta smelt were in the system, although they were not caught in the trawls. So that is very, very exciting for eDNA. And so in development, we have more field-based Sherlock assays for quick analysis, and then using Sherlock to detect delta smelt and tidal restoration sites. So as far as conclusions and our next steps, like I mentioned, eDNA is a really robust tool in the monitoring tool belt, and it can help contribute towards providing the best available science for decision making, and it improves monitoring ability. It's efficient and non-invasive, and we are at DWR are really focused on incorporating eDNA into long-term monitoring and management programs. And one thing I do want to mention is that additionally, we are testing and investigating autonomous eDNA samplers. So essentially water sampling robots that can be out in the wild collecting DNA and either storing it or analyzing that DNA in real time. And so we are trying to use all possible modes of technology moving forward. And one last thing I want to leave you with is our next steps. And so Melinda Bearwald and Daphne Gill have developed a DWR environmental DNA strategy. And what this is essentially showing that DWR sees the advantages and the utility of eDNA. And they have developed this strategy to support water resource management with these key components of forming collaborative partnerships, working with management to prioritize eDNA work, and identifying best practices for effective communication with management. So we're really excited about being able to roll out the DWR eDNA strategy and to help try to implement more eDNA studies at DWR. And so with that, I just want to thank my collaborators at UC Davis and at DWR, and then additionally, the U.S. Bureau of Reclamation, U.S. Fish and Wildlife Service, and at Kramer Fish Sciences, Genadax. And with that, if there's any questions, I'd be happy to take them.
Thank you, Sarah. If you have questions, you can unmute. We also have a hand up right now from Shahiner Islam.
Yeah, thank you. Sarah, great talk. It's Shaheen from UC Davis. So
Hi,
right now
Shaheen.
I am into meta-analysis and review synthesis work with both eDNA and eRNA application in the real world, more like in field-based study, you know. So what I have found from recent literature, then they have been they have bunch of like field-level DNA sequencing technology, and they are getting the great sensitivity, like the way people are processing their eDNA sample in the lab as well. But so far, I haven't found that much literature regarding eRNA work in the field. Like people, I'm not sure, I'm not aware whether they have a fieldable eRNA sequencing or processing their eRNA sample in the field, because you know that eRNA they degrade more quickly than the eDNA. So I was wondering whether your Sherlock tool, whether it's applicable to apply in the field as well, whether you would be able to do some sort of like quantitative, getting some quantitative data from the field, or otherwise you'll have to brought your sample and process in the lab with your Sherlock. And I'm not sure, maybe you know better, you have more more in-depth knowledge, whether is there any other tool they could process eRNA sample in the field?
Yeah, thanks for that question, Shaheen. I know that Emily Funk and Aviva Fisk at UC Davis and GWR respectively are currently working on this, on trying to not only get actual filter samples from the swamps where these nutria are found, which is very difficult to pump that water. And so they are working on trying to kind of get past some technical difficulties with that. Yeah, with the actual sampling. And then I think the goal with Sherlock and with eRNA is the thought that it could be conducted out in the field. And so we have the this Axon machine that can be run on a battery, and we can run Sherlock reactions right out there. It would take an hour or maybe 2 at most. And so I think that would probably be one of the best ways to try to move forward with this eRNA in the field.
We have a question in the chat. How broad an area can a single sample represent?
That is a really good question. And the thing that that my most often answer that I go to with eDNA is that it depends. It depends on the environment that you're in, the type of how much sunlight is in a system, because that can degrade eDNA and eRNA as well. But I know that there are studies, and Susie, correct me if I'm wrong on this, but I think that the distances can range from like a couple meters to like a couple kilometers.
Thank you, Sarah. We have a hand raised. James Hoffler.
Yeah, can you hear me okay? I just unmuted my microphone.
Yeah.
I see you nodding yes. Okay. So regarding eDNA in rivers, seems to me there's some interesting difficulties with that, because if you took a water sample at point A in a particular stream or river, and you get a really big hit, say for Chinook salmon, right?
Mhm.
Boom, definitely there. But you can't really tell if that is one big fat salmon carcass 3 yards upstream that you didn't see, but you got some eDNA from it, or if it's 40 Chinook salmon a mile upstream. Is there any way to deal with that type of problem?
The best way to do that is to have to sample in triplicate or replicate, and then to also have really standardized sampling across your system. And so when I worked at the Department of Fish and Wildlife up in Washington, we would regularly sample like every kilometer. And so we could get then a really good just kind of broad view of the system and be able to detect what is going on. And then if there was any instance where, like, if we did detect a sample or a detection, and we and it was like the only one or something, then we would go back to that system and then do much more intense sampling there, especially for invasive species. But more to your point as well, there's a lot of work being done with correlating the DNA concentration with biomass. And I am not exactly sure of where that is, where that has kind of landed right now. And again, Susie might know more. But I think that's one of the kind of the goal for eDNA researchers is to be able to get that relationship between concentration and biomass.
Okay, so I think I understand therefore that there's no way with eDNA to tell one individual from multiple individuals in the sample.
True. Yeah, and
Okay, all right.
because most of the cases, the assays that we're using are mitochondrial DNA, and so it just can detect that that's the species that you've picked up. There are more sensitive techniques like microsatellites or SNPs where you can get down to the individual, and I don't know if that
Well,
has
that's a matter of touching the fish and taking a piece of the fish, right?
yeah
Yeah,
yeah.
okay. So I would think that a good way to do an eDNA study in a stream, kind of taken off of your idea about one sample every kilometer, would be on a particular day, all at the same time, have X number of people spread out along the creek and get all your samples all at the same time on the same day. And then you can say, okay, well, we got a hit at that, lower in the river, boom, there's a fish. Somewhere upstream from there, there's a fish. You get another hit a quarter of a kilometer upstream, another hit kilometer upstream, and then no hits above that.
Mhm.
So then you've got a pretty clear picture that in that stretch, 2.5, 3 kilometers, whatever it is, yes, there were Chinook salmon there, but not upstream from there.
Yeah,
So,
and in addition to that, you could also do it paired with electrofishing in order to get a even more quantified idea of the number of individuals present and again that eDNA concentration.
yeah, so yeah, just the cost and the coordination for
Yeah.
zapping those fish. And
Yeah.
yeah, I do a lot of volunteer work with Friends of Auburn Ravine. Auburn Ravine flows from the town of Auburn down through Lincoln out to the Sacramento River, about 34 miles. And we have actually heard from our salmon. Now, just so you understand, we don't talk to the salmon. That would be crazy. But we do listen to them, and they live in mortal fear of that shocking thing. So,
I can imagine.
yeah. All right. Thank you very much. That's very helpful.
Thank you.
And we have another question in the chat. Can you speak to sampling techniques, chain of custody requirements for eDNA samples, and can you share the cost per sample for qualitative eDNA results?
Okay, I'm gonna need to read that question. Okay, so sampling techniques. So we currently use the Smith Root eDNA sampling backpack or pump. And that is a really good system because the filters that you get from them are enclosed, they're sterile. You don't have to worry about contamination as much. They're a really great, just all around system. They are fairly expensive though. They're about $15 or so per filter. There are other types of filters as well that you can use with peristaltic pumps. And then you can also collect grab samples from a Nalgene, a sterile Nalgene, take that water back to the lab and then filter it. And then with chain of custody, I guess I don't quite know. Wonder if you can elaborate more on that. And then I also don't know, at least in our lab, like what the cost.
They're in the shop.
I can drop a link in the chat if that's helpful. We have an eDNA primer we put together for the Marine eDNA workshop last year or so, 2 years ago now. And it has a rough overview of some costs. So I'll drop that link in the chat. And for chain of custody, yes, I'd be curious in particular what the question's in regard to, but we follow a chain of custody much like we do for any of our environmental samples. And so especially if there's going to be, if they're going to be used for informing any management action, then I know that would be a critical consideration.
I'm just wondering, thanks, if these need to be on ice right away, that kind of question. Maybe I should have asked that. Thank you.
Oh yeah, great question. And Sarah can weigh in on what they do for their Smith Root samples, and it will often depend on how you're collecting your sample. And so there are some options for filters that are desiccating, and then you pretty much can collect it, and then it dries out by itself, and then it's pretty shelf stable for a period of time. Otherwise, we use some preservation solutions to kind of extend the shelf life of our samples and before we can get them into the freezer, where then they can live at -80, basically for eternity, more or less. But Sarah, go take it away, Sarah.
Yeah, and with the Smith-Root filters, we use the self-desiccating filters, and so those can just stay at room temperature for a couple of months before you can go ahead and extract them. And then I also have used desiccating beads, and so collect the sample on a regular filter, and then put it in a little whirl pack with desiccating beads, and then it dries it out. And again, it's good for a couple of months at room temperature, and then DNA is extracted from that.
Okay, we have another hand raised, Bob D.
Okay.
Hi, can you hear me?
Yes, I can.
Yes.
Excellent. I want to follow up on the river question. I'm in Santa Cruz. Our particular river is not scheduled for any DNA work, although I think a snapshot of biodiversity could be terribly helpful. In doing that, one, does that make sense? Because if I have a desiccated filter, I think I've got that snapshot that will stay analyzable or can put it through an assay for a great deal of time. Could you address using eDNA? And I know it's more of a meta question rather than a qPCR question. Can you address getting your DNA samples simply to put yourself in a position to analyze, compare, and contrast downstream? Thank you.
Yeah, so I think you're asking about if storing samples and then being able to analyze them years later. And I think that is a possibility, although like I said, with the desiccating filters, that it's really best to extract them within a couple of months. And then that DNA can be stored at -80 in perpetuity. And I know that there have been several studies where, I think from Rocky Mountain Labs, where they had collected DNA, eDNA, with the purpose of one target sample, one target species, and then several years later, they went back to that DNA sample and analyzed it for, I think, pearl shell mussel, and so completely different taxa that they were searching for, and they found it in that eDNA sample. And so it just kind of shows that these eDNA samples are gold mines as far as not only tracing biodiversity over time, but also questions that you may not even have, or right now, the things that can just kind of pop up in the future, and you've got this gold mine to go back to. I hope that answered your question.
It does. And Rocky Mountain, I understand, as a matter of custom, stores your samples on site. I think they're in Montana, merely as part of their process for using their service. Will that be true also of the Davis lab that is being put together? And will that lab entertain sort of external non-educational submissions?
Currently, we are only doing DWR projects, and I don't know what the future holds for outside projects.
Thank you, Sarah. We have another question in the chat. And it's about establishing absence determinations. The question is, many threatened endangered species protocols are designed to determine presence absence. We've discussed applicability towards documenting presence of species, but are there examples you could point out trying to establish absence determinations?
That is so difficult. And that's a really good question. I think, again, that kind of leads towards our work with the Nutria, using eRNA to try to determine how well an eradication effort has progressed. And so that is one method to determine presence and absence. And then also just frequent sampling and multiple sampling. Susie, do you have anything to add on that?
I would say that's a tough one, and we're always in consideration of potential false positives and false negatives, and so all the best practice techniques, as Sarah was just mentioning, sterile technique, replicability, sampling at different time points in a year, making sure that there's no inhibitors present in your sample that give you a false sense of nothing being there. So there's a lot of elements of sampling and analytical design that we implement to lower our risk of false positives and false negatives. And then that just increases your confidence that your result, whether it's positive or absence, presence or absence, is a true one. And so it's just increasing the confidence to be able to make those determinations.
Thanks, Susie.
Okay, we have 2 more hands raised. First question comes from Wan Liu.
Hi, this is Yuan Liu from Contra Costa Water District. And you mentioned that the next step is to study this kind of eDNA or eRNA method using the tidal system like in the Delta. And I was wondering what kind of plan is there? And you mentioned like Delta smell, the potential entrainment into the cliff core forbate. How would you design things to detect use eDNA to detect, oh yeah, the Delta smell is there in the system and will be entrained? Yeah, that would be helpful. Thank you.
I'll answer the last question first. And so the goal, or I think one of the ultimate goals, is to be able to use the autonomous eDNA samplers kind of as a first defense of detecting delta smelt DNA in the system as they kind of get closer to the pumps. And so they could help alert us when smelt are moving into the system, and then we could potentially change the water pumping schedule from there. And then I must admit that for the question about the delta smelt for the tidal restoration, that is one of Ravi's goals, and I'm not quite sure exactly what he wants to, or what his goals are for that.
Thank you.
Our next question comes from Catherine Mandela.
Hi, yeah, I am the scientist in the Bay Delta region dealing with primarily quagga and zebra mussels, and I am curious as to I have 2 questions actually. One of them is regarding a handheld CRISPR unit that I remember seeing, gosh, probably over 5 years ago now. I'm wondering, is there a unit, a field unit that is relatively affordable and also that can give you the analysis in the field like within 30 minutes is what I remember this unit say. I don't remember the company that manufactures this one, but I'm wondering if you're familiar with this one, if it's, what its efficacy is, and if you would recommend it. And then let's answer that question first, okay, and then I'll go to the second one.
Okay, yeah, so we use the it's called the Axin, AXXIN, and it can hold 16 samples at a time, and it's battery operated, and we are, so you can take it out into the field, and we are currently working on testing having assays that are pre-frozen, and so you can essentially just extract DNA out in the field. There are many kits where you can extract filter eDNA filters for DNA out in the field, and then you could just add a couple microliters of DNA to your reagents there, and then in half hour to an hour you could have a result. We're also working on testing out lyophilizing reagents, so having them freeze dried so that you would then just have to add water in your DNA, press go, and that axon has readouts in the field on the screen, and so you can see whether or not your sample was positive or negative for your species. And then you can also download that data onto a laptop, and it will show the actual graphs of the fluorescence units. And so we love it, and I cannot remember the cost right now, but I think it's fairly affordable. But it's such a great system.
Okay. Maybe if I can connect with you offline, that might be nice.
Of course.
And then, yeah, lastly, I'm curious if you have since these units can kind of do a, biodiversity scan of what's out there, is there a interagency like spreadsheet that is accessible for all of us to, so that, all the agencies, since we're, a lot of us are duplicating efforts, we know that, but if we could kind of be able to glean off each other to see what everyone's finding, so we're not going out into places that we know a species has already been detected.
Yeah, and I don't know of a spreadsheet of sort, but I know that one of our goals is to be logging all of our data as much as real time as possible into EDI, Environmental Data Initiative, I think. And so that is something that we'll be working on in the future. But yeah, that's a really good question, and I kind of want to go and take a look to see if there is some kind of database of what is actually going on with other studies in the Delta.
That'd be super. Thank you.
I encourage you all to take a look at the chat. There's been lots of posts there. Thank you, Susanna, for putting stuff there. Susie's been great at putting in information also from the Molecular Methods Working Group. And if you haven't participated with that working group under the Monitoring Council, I encourage you to visit their website and attend one of their meetings. It's one of the better working groups that we've seen emerge recently. Do we have any other questions? We're coming up on 12:30. Thank you, Sarah, for providing this excellent Q&A opportunity.
No problem.
If there are no other questions from the group, I had one last question, Sarah. It was so fun to hear about all of this. And I was curious if you have like an approximate timeline to develop a new CRISPR or Sherlock assay, if we had a new target species of interest.
So I think, yeah, work is currently being done on the zebra quagga mussel assays. I want to say, yeah, a year.
A year. Okay.
Or more. Yeah, we've got so many projects right now.
Yeah.
But that's in collaboration with UC Davis as well. And I know that we like for different assays that we can take from kind of modify qPCR assays, those from those sequences, and then use them for Sherlock development. But it takes quite a bit of work to make sure that, again, that you're only amplifying your targets and that it's as sensitive as possible.
Got it.
Yeah, like Sean Canfield mentioned in the chat, that some Sherlock assays are easier to design than others.
Got it. And is there a length consideration? Obviously, with like qPCR, digital PCR, we're like winnowing in on a length, which can sometimes be limiting. For CRISPR, are we like really short, or is it kind of medium?
I might defer that to the chat for Sean.
Okay, great. I'll follow up. I'd love to learn more. But that's exciting to hear that sometimes it can be, it sounds like from Melinda, 6 months for a new design. So that's great to hear. Awesome. Okay. Great. That brings us to,
Susie,
almost 12:30. Perfect.
Yeah. Before we break, there was a request for access to the presentation. So Sarah, if you can make a accessible PDF, I can post it on the Collaboration Network's webpage. Otherwise, maybe Susie could host it for us.
Yeah, I can make a PDF of it.
Perfect.
Yeah, thank you.
Great. Thanks so much, Eric, for having us as always.
Oh, my pleasure.
And
Thank
thank
you,
you, Sarah.
Susie.
Thank you.
That was so
Thank you,
fun.
Sarah.
Awesome. We'll hopefully see you all at the next webinar and be on the lookout for when that gets scheduled. Thanks, everyone.
Thanks.
See you
Bye.
next time.
Have a great day, everybody. Thanks again.